Hello everybody. So today we are continuing the study of proteins, specifically with respect to their structure, or in other words the levels of organization of proteins. The three-dimensional shape of a molecule is referred to as conformation. That three-dimensional shape or the conformation is responsible for specific biological role of a protein. Under classification, we have classified them based on the biological role as enzymes, antibodies, hormones, structural materials, transporting agents, and so on. Those specific functions are due to their specific three-dimensional shape or the conformation. So once they are not in that particular three-dimensional shape, they cannot exhibit their biological role. So the biological function or their biological role is due to their three-dimensional shape. And the native conformation of a protein is that conformation in which it is biologically active. So whatever form it will be when it is doing its function, that is referred to as the native conformation or the three-dimensional shape. So depending upon the complexity, proteins may have four levels of structure. The primary structure, secondary structure, tertiary structure, and quaternary structure. Primary structure refers to the sequence of amino acids. The order in which the amino acids, so in the chain the amino acids link to one another, this sequential order, arginine, cysteine, proline, methionine, leucine, aspartic acid, glutamine. For example, if this is the arrangement, only when this particular arrangement is there, then only the protein can function or exhibits its biological role. This orderly arrangement or sequential arrangement is referred to as primary structure. So for three-dimensional shape of protein or the three-dimensional structure or the conformation in which it exhibits biological activity, the amino acid sequence is very important. That is referred to as the primary structure. When there is any change in the order, in the sequence, it cannot exhibit its biological function or it loses its biological role. It is just a protein, not with any specific function. Thereby, primary structure is very important for protein. One example we can quote here to show how primary structure is important. Hemoglobin, it has four polypeptide chains, alpha 1 and beta 1. In one of the beta chain, in one of the polypeptide chain, due to mutation, glutamic acid is substituted by valine. So during its synthesis, it undergoes mutation and glutamate in one of the beta chain, glutamate in place of glutamate, valine occupies its space. Because of the change in the sequential order, it cannot perform its normal function. What is the role of hemoglobin? Transportation of oxygen. Oxygen will bind to hemoglobin thereby it is carried to different cells. That absorption of oxygen, there will be a disorderness, and oxygen cannot absorb hemoglobin properly, thereby the cells acquire a sickle cell, they become sickle cell, and hence this disorder is referred to as sickle cell anemia. Just due to change in one amino acid in one of the polypeptide chain, it leads to a disorder what is called as sickle cell anemia. So in that way, primary structure is very important with respect to biological function. Nextly, secondary structure. Secondary structure refers to orientation of the polypeptide chain in space. The spatial orientation. So if it may be a fibrous protein or globular protein, whatever it is, how the polypeptide chain remain in space, it may have a helical, it may remain in a helical form, or it may remain in a beta pleated sheet structure. There are several forces which stabilize this, for example, hydrogen bonding and so on. It's something like an asbestos sheet arrangement that is referred to as beta pleated sheet structure, or this is alpha helical structure. The polypeptide in space, it may remain in a coiled form or a helical form or a sheet or beta pleated sheet form. Mainly hydrogen bonding is responsible for this. So that refers to the secondary structure of protein. Tertiary structure is, if it is a globular protein, there will be a folding of the polypeptide chain several times. The protein molecule folds itself several times. What are the forces which stabilize the protein to remain in such a globe-like structure or global form or in a spherical structure? The folding of protein molecule and the stabilizing forces which make the protein to remain in that particular form is the tertiary structure of a protein. Quaternary structure is for oligomeric protein. Oligomeric protein means protein with two or more polypeptide chains. If a protein has two or more polypeptide chains, for example hemoglobin. Hemoglobin has 4 polypeptide chains, alpha, alpha 1, beta, beta 1. How all these 4 polypeptide chains are coordinately linked to one another, how they remain in one form, as a whole it remains as one protein molecule. Though there are 4 polypeptide chains, altogether they are coordinately linked to the metal atom iron. In that way they remain as a whole as one protein molecule. So for oligomeric protein, it's the quaternary structure which determines how the 2 or more polypeptide chains are coordinately linked to one another and remain as one system, and that is again responsible for their particular function. Yes, to get more clarity about the secondary structure, tertiary structure and quaternary structure of proteins, you can see here the alpha helical structure of a polypeptide. These are the hydrogen atoms and the red ones are oxygen. The orange color is nitrogen and the blue color is the side chain in the amino acid, and the black ones are carbon atoms. Say in between oxygen and hydrogen, see red one is oxygen, yellow one is hydrogen. There is a hydrogen bond. This hydrogen bond in the protein molecule leads to that particular conformation. This hydrogen bonding is responsible for the protein molecule to acquire the coiled structure. It coils due to these hydrogen bondings. So this will be the orientation of the protein molecule in space, or the spatial arrangement. That is nothing but the secondary structure. The same thing is depicted here. So here you observe the individual atoms in the amino acid. So here it is just a polypeptide chain, and these black dotted lines are the hydrogen bonds, and these hydrogen bonds are responsible for the polypeptide chain to remain in the twisted form or the coiled form in space. So this is the secondary structure. Here you can observe that what tertiary and quaternary structure of a protein means. So this is a myoglobin like hemoglobin. Hemoglobin has 4 polypeptide chains. Myoglobin is a single polypeptide chain. However, this myoglobin will not remain in a thread-like structure. It is a globular protein. It folds several times and remains in a spherical form. What is responsible for this folding? What are the forces which stabilize the molecule to remain in such a form, in the folded form? That is tertiary structure. So what are the forces responsible for it? These are the stabilizing forces. Hydrogen bonds again. So this is the beta pleated structure, what we referred under secondary structure. You can see sheet-like structure here in the protein molecule. So thereby there is a folding here because of beta pleated structure. These red colored dotted lines are hydrogen bonds. Within the protein molecule there is helical structure also, coiled form. Here again you observe hydrogen bonds responsible for that coiled structure. So within the protein molecule in the polypeptide chain, you observe both beta pleated sheet structure as well as the coiled form or the helical structure. But for both hydrogen bonds or the forces or the linkages which makes them to remain in that particular form. So hydrogen bond is one of the force for folding of the molecule or stabilizing force. In addition to that, hydrogen bonding between R groups. See here there is aspartic acid or glutamic acid which has carboxylic group, COO-, and this may be tyrosine with OH group. So hydrogen bond between oxygen of the carboxylic group and hydrogen atom of the phenolic group or hydroxyl group. So hydrogen bond again between the R group is responsible for folding of the protein molecule or one of the stabilizing force. And non-polar interactions, that is another force. Non-polar interaction refers to the R between the R groups. See, isoleucine, leucine, valine, methionine. Between isoleucine, leucine, valine, you find CH3 groups. In phenylalanine, C6H5, methionine. Between the side chains, there will be some interactions, what we call it as non-polar interaction, which also acts as a stabilizing force for the tertiary structure. Disulfide bonds. These are cysteine residues. In cysteine, the R group will be CH2SH, CH2SH. So between SH group with elimination of hydrogen, these sulfur atoms have been linked. So between the cysteine residues, between the sulfide groups, such a bond formation occurs, what we call it as disulfide bonds. This is another force, stabilizing force for the tertiary structure. Coordination between metal ions, that again is one of the stabilizing force. Ionic interactions, let this be lysine. Lysine is in the side chain CH2, 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 NH2. NH2 remains in NH3 plus form. This may be aspartic acid or glutamic acid with carboxyl group. So it ionic interaction means interaction or bond between positively charged and negatively charged R groups. Aspartic acid and glutamic acid negatively charged, lysine is positively charged, so there is an interaction between the two R groups. That is another force which is responsible for folding of the molecule or force stabilizing the tertiary structure. So in that way, the force stabilizing the tertiary structure are hydrogen bonds, non-polar interaction, disulfide bond, coordination with metal ions, ionic interaction. All these forces stabilize the tertiary structure or makes the molecule, protein molecule or the polypeptide chain to remain in this form. For quaternary structure, so quaternary structure is for oligomeric protein, proteins with 2 or more polypeptide chain. Here hemoglobin has been taken as example with 4 polypeptide chain, alpha 1, alpha 2, beta 1, beta 2. So they will not remain as free polypeptide chain. All the 4 polypeptide chain, alpha 1, alpha 2, beta 1, beta 2, they are coordinately linked to one another, and hemoglobin in that way remains as one protein. So what is responsible for this? It's the metal atoms porphyrin ring system within the molecule which links the 4 polypeptide chain coordinately and helps the molecule to remain as one system. So that is quaternary structure of protein. So the specific functions or the biological role of proteins is due to their 3-dimensional structure or the native conformation. The 3-dimensional shape or the structure refers to the native conformation. So once they are in native conformation, they can perform their function, they exhibit their biological role. If the forces stabilizing that particular 3-dimensional form, if that is, when the forces disrupt these structure, the 3-dimensional shape, then they cannot perform their function. So disruption of the native conformation of protein is referred to as protein denaturation or denaturation of proteins. Denaturation of protein refers to loss of 3-dimensional shape or structure of protein and hence the biological role. Once they lose the 3-dimensional shape or structure, they lose their biological function also. They cannot perform their usual function. Then they are said to be denatured, and this phenomena is known as protein denaturation or denaturation of protein. So the factors responsible for protein denaturation. See, we have referred to several factors which stabilize the tertiary structure and other levels of organization of proteins. So if any factor which destabilizes that will be the factor responsible for denaturation of protein. Electrolytes, acids or bases. Addition of acids or bases or in electrolytes leads to protein denaturation. Organic solvents or detergents also denature protein. Mechanical agitation. Even when the protein solution is shaken vigorously, that also leads to denaturation of protein.
Sir.
Reagents such as 8 molar urea, beta mercaptoethanol. They also denature proteins. Even heat, when it is excessively heated to a high temperature, all the forces stabilizing the three-dimensional structure is lost, hence it gets denatured. So these are the factors contributing for denaturation of protein. Once it loses its three-dimensional structure, it cannot function, it cannot exhibit its biological role, then it is said to be denatured, or phenomena is denaturation of protein. Bringing back to its native form. The protein is denatured because it is not in native conformation. If you bring back to its native conformation, if regains its three-dimensional structure, it can start performing its function. That is referred to as renaturation. That is called renaturation. Loss of biological activity due to loss of three-dimensional structure is denaturation. Regaining the biological role through regaining its native conformation is renaturation. There is one experiment by Anfinsen, Christian Anfinsen, conducted this experiment to show how a denatured protein can be brought to its native form, or in other words, it can be renatured. That is referred to as Anfinsen's experiment. He took ribonuclease to perform this function. Experiment 1 is denaturation. Experiment 2 is renaturation. Experiment 1 is denaturing it. Experiment 2 is renaturation. Ribonuclease is a single polypeptide chain with 124 amino acids. It has 4 disulfide bonds. So this is one of the major force stabilizing its tertiary structure or the three-dimensional structure as a whole. This was treated with 8 molar urea and beta mercaptoethanol. By treatment with 8 molar urea and beta mercaptoethanol, all these 4 disulfide bonds were disrupted. Because of disruption of these bonds, it loses its three-dimensional structure and remains in a denatured form, or it is denatured form. This is native form. Now it is in denatured form because of disruption of this disulfide bonds. It is no more in that particular three-dimensional structure. This cannot perform its function. How this denatured can be brought to native form? Further, this denative form was subjected to dialysis. Through dialysis, whatever 8 molar urea and beta mercaptoethanol were there, the reagents, the denaturing reagents were removed. Through dialysis in a suitable medium, those reagents which were responsible for the denaturing, that is 8 molar urea and beta mercaptoethanol, were removed from the medium. Thereby, again, those 4 disulfide bonds were introduced in the molecule, thereby it regained its native form, and now it is biologically active. This is referred to as Anfinsen's experiment, where enzyme ribonuclease was taken, first subjected to denaturation using 8 molar urea and beta-mercaptoethanol. So thereby the enzyme was left over in denatured form, inactive form. Further, the denatured one is subjected to dialysis to remove the reagents which were made use. So thereby it regained its native form and became biologically active. This experiment is referred to as Anfinsen's experiment, and for which in 1972 he got Nobel Prize. So this shows the importance of the three-dimensional structure of proteins, how it is responsible for their biological role or biological function. With this, I conclude the study of proteins. Whatever it's there in the syllabus, in your syllabus, that has been explained, and in another part I am going to send the photographs which will support and which gives more clarity about what all you have learnt in part 1 and part 2. Thank you very much.